This is episode 11 of the Immunology Podcast, Adoptive T-Cell Transfer with Dr. Christopher Klebanoff. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rao. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. Today we have Dr. Christopher Klebanoff from the Memorial Sloan Kettering Cancer Center on the podcast to talk about his work adapting adoptive T-cell transfer to patients with solid malignancies. Well, we've also got our usual roundup of recent highlights in immunology news coming up. But first,
STEMCELL Technologies would like to introduce you to human immunology news, covering everything from immunotherapy, autoimmunity, and adaptive and innate immunity. Human immunology news keeps readers current with the latest news, research, policy, events, and jobs relevant to the immunology community. Subscribe for free at www.humanimmunologynews.com.
Hey, how are you, Brenda?
Hey, Jason, doing great. How about you?
Oh, living the dream, trying to get a little bit of summer in. You going anywhere by chance?
Well, I am, because the summer ain't coming to the lowlands, so I'm going to Spain in a couple of weeks.
Oh, nice. I'm heading out to Long Beach Island in a bit for a little bit of time at the beach. But of course, I'll bring you papers with me to read, because what else do you do at the beach except read papers? Hit me with what you read at the beach last week.
So, first of two papers, as always. I'm going to go a little bit T-Rex friendly in my papers today. And one of my papers is going to harness T-Rex for good. Another one is getting rid of T-Rex by targeting specifically. Okay, so first paper, dual targeting of CTLA4 and CD47 on T-Rex cells promotes immunity against solid tumors. So we know T-Rex in tumors are bad news. So this paper from Science Translational Medicine, first author Anli Zhang and Shenwan Ren from the lab of Yang Xing Fu at UT Southwestern in Texas. They are looking at what can you do about T-Rex in solid tumors. We know that they are associated with poor prognosis, and we know that there are ways of depleting them using antibodies. And there's a lot of controversy whether, famous checkpoint inhibitor CTLA4 actually works by inhibiting T-Rex that express high amounts of CTLA4 on their surface. And other molecules that are expressed in T-Rex can be targeted, but we know that depleting them can result on side effects and immunity, autoimmunity in other places. So it's very, very difficult to target T-Rex. And so, as I mentioned, CTLA4 is a checkpoint inhibitor that has been used in the clinic with mixed results. Can have very activated immune system in a very strong manner, but that often results in adverse events that are a little bit prohibitive sometimes. And we don't really know, there's still a little bit of uncertainty whether this CTLA4 inhibition is mainly targeting the effector cells and reducing their inhibition, or is actually depleting T-Rex by binding to them and starting antibody mediated cell depletion. So they're in this paper, they're looking into a molecule, trying to design a molecule that can try to, on the one hand, take advantage of the fact that the cells that have the highest expression of CTLA4 in tumors is often T-Rex, but without having such a high affinity that then they just bind everything, and also they could also bind T-Rex that are not in the tumor, that are also expressing CTLA4 to a lower extent. And they do this by combining another signal that also is expressed in many cells, which is mediated by CD47, which is a do not eat me signal that binds to what is known as a signal regulatory protein alpha, or SIRP alpha. And this do not eat me signal is usually expressed on cells and prevents or reduces the phagocytosis by, for example, other cells in case of opsonization, in case of antibody binding on the surface of the cell. So what they generate, so there has also been targeted, CD47 has also been targeted by inhibition using particular antibodies and combining them with therapeutic antibodies such as rituximab that will provide an eat me signal with a Fc receptor, and then would, by inhibiting the do not eat me signal, that would increase phagocytosis of the cell. But the problem is that CD47 is also expressed in, for example, red blood cells and platelets. And it's also sometimes hard to get the antibody into solid tumors because usually they're not very well vascularized, or it's hard to get these things into the inside of the solid tumors. So what the idea is that they want to, on the one hand, block CD47, so block the do not eat me signal, preferential Tregs, and provide an eat me signal. So therefore you can, macrophages or other cells in the tumor can target Tregs and deplete them specifically without harming Tregs outside of the tumor, and without harming other cells that are beneficiary to the response. And so what they do is they do a molecule which has an anti-CTLA4 specific arm of an antibody, but then the other arm is replaced by a SIRP alpha binding domain that would preferentially bind to CD47. But neither of these do it with a very high affinity, and only the combination of both molecules generates recognition and triggers phagocytosis by, for example, macrophages. And so they do this and they test how they do a bunch of experiments in mice using different mouse models, MC38, which is a color cancer model for mouse tumors. And they show that by treating this, they can specifically target the Tregs in the tumors that have enriched expression of CTLA4 compared to Tregs outside the tumor. And the fact that they use, instead of using an anti-CD47 antibody that binds the binds CD47 very strongly, they use the SIRP alpha domain. This, together with also weaker CTLA4 binding, generates a very nice and specific binding only to double positive cells, which in this case will mostly be the Tregs. So it's a very nice, mouse models. They do test it in rat knockout mice, and they see that they only can see this effect when there's T cells involved, so by depleting Tregs. And they show that there's a loss of Tregs in the tumors. And they also, I think it would be a nice platform to see if that can be applied into human treatment.
Well, that's really neat. And so from a clinical perspective, it sounds like it's a bispecific antibody, which means it has two arms, which is actually a pretty common thing going through the clinic right now and through, and by clinic, I don't mean in patients, like FDA approved, but a lot of work's being done that in pharma land on bispecific antibodies. So it's kind of the next generation after these initial monoclonals with one binding site. So it's interesting to see this application. And Tregs, I predict in the future we'll be talking about Tregs a lot, Brenda.
Me too. I have that feeling. But I just want to say it's not really a bispecific antibody, because one of the specificities is actually the binding domain of that binds to CD47. So it's not really an antibody. It's the part of the receptor that interacts with CD47.
Oh, so it's
I think it's also even kind of a half done bispecific molecule / other, like, I don't know,
pseudo receptor sync. Yeah, that makes sense. Yeah, that's like Embro works on as a TNF receptor. Okay, interesting. Now that's it's cool seeing what they're starting to come up with. And you're kind of seeing that now that we have this proof of concept for some of these, there's a lot of like iterative adaptations where I think we'll see a lot of therapy coming out soon. All right. Well, we'll play our usual game. Is this paper about our favorite virus or not? It is. A high throughput cell and virus-free assay shows reduced neutralization of SARS-CoV-2 variants by COVID-19 convalescent plasmid. First author is Craig Fenwick, and last author is Didier Touronneau, and it's in Science Translational Medicine. So obviously, if you can't get this one, we have problems here. But we have another COVID paper. There's two really interesting things I think about this paper. One is kind of the higher level thought. So this paper came out August 4th, and it talks about the variants of concern as the alpha and beta variants. Which, because if you think about how long it takes to publish something and get through the process, they probably did this during that time frame. So this just goes to show you, for those who are more maybe new to science or trying to join this podcast to learn about things but aren't necessary in the trenches. This work was probably done 9 months ago, and now it's out, and so that's why you're seeing a lot of this stuff on preprint. But still, now we're already up to Delta, but the points here still apply. And this paper can be pretty quickly described as we're trying to figure out if a person who has neutralizing antibodies, how that works against various variants, right? Or do you have neutralizing antibodies? And the problem is these assays can be tough to do because you have to have good viral particles often, is how the other assays work. The lentiviral-based system, as they point out, doesn't work very well because of where the spike protein is made in a lentivirus system is on the plasma membrane, but coronaviruses do it in the Golgi complex, an endoplasmic reticulum. And so you don't have the same protein folding package. So it doesn't look quite right to the antibodies and everything else. So you want to go to something cell-free as a goal, and can you get a good system that can show, these antibodies in this person neutralize the effect of the spike protein on the ACE2, the angiotensin 2 receptor, right? Because that's what the spike protein binds to. And so they went with a cell-free assay. It is pretty well done here. What they did is they took Luminex beads and had those have the, to try to describe this right. So they took a Luminex bead, which is nice because those Luminex beads can be detected in flow later on. And they put that with a spike protein trimer, the full form, not just the monomer. They did the full trimer, which is part of their technology. Then to that, they add a ACE2 that has a mouse FC tag on it so that you can use an antibody against the FC tag for secondary detection. And so if you have fluorescence antibody, then you can detect that. So in a regular system, you take the Luminex spike beads, you mix it with angiotensin, the ACE2 recombinant, and you'll see binding, right? Because you'll then then you put the antibody on, it flows. And then you convert this to a quench assay or a neutralization-based assay, because if you add serum from a patient who's been exposed to COVID, then those antibodies will bind the spike protein and prevent the angiotensin 2 fraction from binding to it, which means there's no fluorescence. And so because it's Luminex, they can have different wells you can have different beads with different spike proteins on them for the different variants that can all be read at the same time. And so you can then multiplex this. This paper basically shows they created a multiplex Luminex-based cell-free assay that works for detecting neutralizing antibody function because the antibodies will block this interaction pretty reliably. Now, it has a lot of advantages over cell-based things. Like, this is way better. I think the disadvantage still is it still requires recombinant proteins, which of course anything will, but that's still going to be somewhat expensive, but it's still a heck of a lot faster than anything else you got. They use Chinese hamster ovary cells for the spike protein. That's pretty easy to get. I don't remember how they did the ACE2 recombination, but they can pump it out. So I think this has a potential, especially if they can scale it up and produce large quantities, to be a really good assay. So I thought it was really cool. It's more of a technology paper, but I've gotten more and more assay development in my life recently. I thought this was really cool to see. And I also thought it was a really interesting point that they really validated against the alpha and beta variants. And we're already on to Delta now, our favorite letter of the month.
Okay, what happens when we make it all the way through the Greek alphabet?
You spike the football and we're done. We just don't care anymore. No.
That's done. Then we just give up. Yeah. We don't count them anymore. We're like, This is it. That's all the Greeks intended. So let's just leave it to rest. Yeah, this is very cool because then it would allow for maybe more commonly evaluating the neutralizing antibodies in patient serum without having to go through all the hassle, right, of doing cell-based assays.
Yeah, no, it's really neat. And it uses this existing platform, which is smart too. Luminex is a thing that exists and can be adapted for all types of different antigens. It's usually if you think about cytokines, but like I was really impressed with the approach. It was a smart design.
The Luminex platform, can you just quickly describe
so you
it?
have beads that you attach stuff to, that then you measure binding to that bead of something. So you could have like, and then some type of sandwich-based kind of ELISA system, but it's bead-based, and so those beads can have fluorescence on it and go through a flow. But because you have different beads, you can gate on 2 colors at a time. One color being the bead color, and the other color being your detector. So you can go, of the bead color, how many green? Of the next bead color, how many green? And so forth. So you can like do 18 cytokines at once or something like that on Luminex platforms.
Yeah. So you can
Because you
do
can mix,
very
you have all the
multiplexing.
beads in one well of the different types, and then you add the sample in.
And then because there are beads, you can actually measure them in a cytometer, right? Because you cannot
You
measure
measure the
just
color of
cytokine molecules.
exactly. You measure the color, and then the secondary output of what other color is that, in this case, the antibody that is bound to the ACE2 FC chimera. So you make that sandwich thing. So it's very common for, like, you use it a lot for high throughput blood screening of cytokines that you've done bad things to mice in.
All right. Then moving to my second paper of the day. This paper comes from the lab of Timothy Han at the University of Pittsburgh. And first author, Amrita Bhattacharjee. And it was published in Immunity, and it's called Environmental Enteric Dysfunction Induces Regulatory T cells that inhibit local CD4 T cell responses and impair oral vaccine efficacy. And in this study, they look into a syndrome that is quite problematic in certain areas of the world where there's severe malnutrition, and which is known as environmental enteric dysfunction. I'm pretty sure you heard about it. And this is caused, as I said, by a combination of chronic malnutrition and chronic infections, and results in stunting of growth in children. And what is very interesting, it also results in a reduced oral vaccine efficacy in children. When they get their vaccinations when they're young, you see lower efficacy rates among these populations. And so when you look at the intestine and the situation there, you see that children with EED have microbial dysbiosis. They have infiltration of lymphocytes. Importantly, they have a flattening of the villi in the small intestine, and this results in malabsorption because there's less area to absorb nutrients, and therefore the stunting and the growth issues. And so the researchers wanted to look, trying to generate a mouse model that would, with certain fidelity, mimic this pathology, and that can be used to understand why is it that oral vaccination is reduced in patients with EED. And so one of the characteristics, of course, of oral vaccines, and the reason why they're can be very good, is that they can induce long-lived intestinal responses, particularly CD4 responses and IgA positive plasma B cells that, by secreting IgA, protect the host from infection from microorganisms that go through the gut. And what they see, so they will look into how to understand the immunity that in the gut that is interfering with the vaccination. So they developed this model in mice by finding a specific strain of E. coli that is invasive, and they show that in mice that are subjected to a malnourishing diet, if they expose the mice to this invasive E. coli, they can generate EED-like symptoms. They can show that the bacteria, they adhere to the intestine, they generate inflammation, they see a reduction in the length of the villi, they see the mice lose weight, and they see decreased intestinal barrier function. Now, these all are symptoms that are consistent with EED in humans. And they only see this at a combination of malnourishment and the introduction of this invasive bacterial strain. So when they have this mice, they validate this model, and then they use it to understand the consequences of oral vaccination. So when they compare mice, EED mice, with regular mice, with non-malnourished mice, they show, so they normally, the immunization with a attenuated heat labile toxin from E. coli usually generates a robust immune response that has a CD4 activation of CD4 cells with a Th1/Th17 phenotype. They also see the upregulation of B cells that are positive for IgA and a protective response against then the infection with the bacteria with an E. coli expressing this toxin. But when they look into the EED mice, this response is blunted. They use MHC2 tetramers to find CD4 T cells that are specific against this toxin, and they fail to accumulate in particular areas in the mouse. So they look into, and what is interesting, they only see this in the small intestine. They don't see differences in the mesenteric lymph nodes. They don't see differences in the colon, but they focus on the small intestine, and that's where really the difference comes. And the EED mice cannot control an infection later with the bacteria producing the toxin, and they see that the composition of the microbiota is different in EED mice, also what's similar to what is seen in humans. And then so when they look at the types of cells that are in the small intestine in these mice, they see one particular group, Tregs. They see that Tregs that are FOXP3 and ROR gamma T positive cells, which are very characteristics of Tregs that are induced in the intestine upon response to intestinal antigens, they accumulate and they increase their numbers. And what they do, they test and they show that it's these Tregs whose expansion is initiated by the inflammation caused by the bacteria, that they are blunting the CD4 response and therefore reducing the efficacy of the vaccination in these mice. So basically, they show that the whole environment in the intestine is modified, particularly the production of retinoic acid. That's probably the reason why there's an increase in the induction of FOXP3 positive cells. They show that ROR gamma T is necessary for these FOXP3 cells to accumulate, which is also consistent with what we already know about Tregs in the gut. And so very interestingly, also they correlate this with results seen in humans in which they see that children suffering from ED have indeed increased amounts of CD3 positive and CD25 positive cells in their intestine, which are probably marking Tregs. So I thought it was very nice model and an interesting view on how Tregs can interfere with the immune response.
Well, that's really interesting. And then, of course, the diet and the microbiome seem to be helping regulate the ability of the Tregs to do their job because, of course, #microbiome.
Team microbiome.
Exactly. No. We're going to make a gut immunologist out of you yet, Brenda. And you like Tregs, right? And Tregs they love the gut and, oral tolerance. It's where you want to be. It's where the action is. All right. Well, last run of the day. Vertically transferred maternal immune cells promote neonatal immunity against early life infections. First author is Aina and Elias Sletzler, and last one is Petra Clark-Ark. This is in Nature Communications, and this was a really interesting paper. So you actually can detect maternal DNA or fetal DNA in a mother during gestation, but there's also maternal immune cells that in a small fraction transfer over to the baby. And these are called maternal microchimeric cells. We know they're there, and they transfer from mother to fetus, and you can even see them for a little bit after the fetus is born. And then they go away because immune cells die. Sad lives. But the effects of them weren't really known. So this group created a mouse model to track this, demonstrated that, and then did a whole bunch of work to ablate different immune cells in the mother to demonstrate what happens in the fetus. And it's cool. So to quickly walk through this, they took the whole CD45.2, CD45.1 system, as well as this the MHC H-2D B versus H-2D D variant of one of the MHCs, and used those to create markers that could track by crossing those mice, so that the offspring were heterozygotes, and they could see those, what came from mommy and what came from daddy. And they could then track the maternal ones properly. So it's super clever, because they could like measure the fraction that was present in the babies to show that they had this, the MMCs were the CD45.2 H2D BB. So basically they were one type, not the other. So they could screen for that. That was pretty clever. And so they show that, and they show they can identify these populations, and they're mostly lymphocytes. And so they kind of do all that work. And then they look at the bone marrow and then do some culturing and show these cells help the bone marrow generate monocytes. So that's the first step. They identify, make a mouse system and show that these cells in vitro help generate monocytes. And then they knock it out. So then they go and they do a RAG2 gamma C knockout mouse. And they're crossing bulb C and B56 mice together. And then basically they have MMC low knockout mice, because if you get rid of RAG and the gamma C as well, you basically lose a whole bunch of your immune. So these are basically T, B cell, and NK cell deficient mice. And so if that's the mommy, then the baby has no MMCs, these maternal microchimeric cells. And then they look and say, oh, look, they have less. The MMCs actually make the baby make more monocytes from their bone marrow. That's what they work, but it occurs in the myeloid compartment, probably after the GM CS, the GM cytokine induction step. So it's later on. They're seeing that it shifts kind of later and makes more monocytes. And then they got the clinical implications of this. So mice with higher numbers of MMCs and monocytes are more resilient against murine cytomegalovirus infection. And then where they really finished this off is they looked at human cord blood and the number of low respiratory infections in the first year of life in babies, and found that the more MMCs that were in cord blood, the less respiratory infections occurred the first 6 months of life. And they correlated very well. And the effect eventually goes off and wanes away. But the number of MMCs that were in the baby determined how many infections they had. And there you go.
It's amazing how many things we get from mom, huh? It's just mind-blowing. I don't think I've thanked my mother enough for all she's given me. Well, there's also those antibodies when I was a baby, those maternal antibodies, and then in the milk. Like, there really is incredible how much of the baby's immune system can be determined by who the mother is and what the mother does.
It's absolutely fascinating. No, it's very cool, and a whole other angle that we hadn't thought about before. I think it's really neat, and they really started getting some mechanism with some good mouse biology and clever crosses to get there, which was neat to see.
Yeah, it was indeed nice and clever and very elegant experiments. I thought I'm very impressed.
Excellent. All right, then. Well, it's time to get on with the show. Decorate your lab with a Nature Protocols wall chart outlining the production of CAR T cells for therapy, from apheresis collection and T cell enrichment to gene modification, expansion, and delivery. Request a free copy of the wall chart at the STEMCELL Technologies T Cell Therapy Resource Center by visiting www.stemcell.com/T-cell-therapy.
We are joined today by Dr. Christopher Klebanoff from the Memorial Sloan Kettering Cancer Center in New York City. The Klebanoff lab focuses on T-cell immunology and has an emphasis on adoptive T-cell immunotherapy. Dr. Klebanoff, thank you so much for joining us today.
Hi, Brenda and Jason. It's my great pleasure to be here with you. Thanks for the invitation.
Thank you for coming.
So maybe we can dive right in a little bit into the research that you guys are doing. So one of your focuses is T-cell immunotherapy, and what can we do with those T-cells? What can we treat, and in particular against cancer, against solid tumors? You have done a lot of work at understanding how do we make the best cells, how we identify the best cells to start these products with, how do we make them stay fit for generating the best therapeutic. So maybe would you like to tell us a little bit, how did you come into this field? You had a lot of years working at the NIH with Steve Rosenberg, Nicholas Restifo, also big people in the field. So maybe would you like to tell us a little bit about how you got here?
Yeah, a little bit of a long and meandering adventure. So I initially became involved in tumor immunology research when I took a sabbatical as a medical student and was looking for a research opportunity. I kind of had grown a little bit uncomfortable with just rote memorization from medical school and desperately wanted to do something a little more creative. So I was very fortunate to be able to participate in a program which has since gone by the wayside, but it was sponsored by the Howard Hughes Medical Institute, and it brought early career medical students to the National Institutes of Health in Bethesda, Maryland. And it was this extraordinary open-ended opportunity. You didn't have to identify a mentor in advance of this program. You could just kind of show up on the campus at NIH and start interviewing around. There's over 1,000 PIs at NIH, so it's just an incredible diversity and density of PIs. And it turns out that because your salary is covered, and because I think there was a reputation that the colleagues who came before me were productive and did well, you could reach out to literally any of those 1,000 PIs, and they would respond pretty quickly, invite you to come through. So at the time, I was interested in cardiology, sort of, cardiovascular medicine. But I really looked at this as an opportunity to kind of broaden my horizons. And at the time, I noticed that there was a research team which was led by Nick Restifo in collaboration with Steve Rosenberg. And at the time, they were deeply interested in developing therapeutic cancer vaccines. And as a medical student, I obviously had heard of vaccines. I had taken courses in vaccinology, and it was entirely centered on protective immunity against infections. And of course, primarily what we studied in medical school in the context of vaccinology at that point was humoral immune responses, generating antibody responses. So to hear the idea or the premise that you might be able to immunize in a therapeutic way a patient with cancer rather than an infectious disease and recruit T cells just blew my mind. So I knew nothing really about T cells at that time, had never heard of a therapeutic cancer vaccine. So I emailed Nick, and he invited me through, and the rest is sort of history.
Oh, that's interesting. I heard of that program. In the past, there was actually kind of like Howard Hughes did a lot of things. I was an undergrad fellow at one point with it. So I wanted to dive in. MD, who does research now, and a lot of research. I did the MD PhD, which is the long, very delayed road to that process. What would you advise for people more on the MD side, getting into research, and how to do it without going back to grad school for a long time? What do you think are the avenues these days? As you mentioned, the Howard Hughes process doesn't exist. Med students can go around and beg to be in the lab, but just going and hanging out in the lab doesn't get you there. So for the people who have realized that there's a lot to do there, any thoughts on how to kind of steer that?
Yeah, so in full disclosure, I am an MD only. I don't have a graduate degree in science. I don't have a graduate degree in immunology, which is a point that I remind all of the junior trainees who rotate with me, who work with me. So at the end of the day, I think it's just like medicine. Science is a practice. And so the only way to develop the practice is to roll up your sleeves and become involved. I think for, more classically trained clinicians who are interested in, all of the exciting advancements that are happening in immunology, especially cancer immunity, just finding a lab, rolling up your sleeves, taking some time out, hopefully at least a couple year period of time, is just a great opportunity to gain experience hip to hip with graduate students and postdocs. And, it's a real truism that everything that is written down in a textbook either is demonstrably incorrect or the paradigms change on a relatively short time scale. So this is my bias, and I think graduate programs are fantastic because of their structure, but it's not the textbook learning that really is what someone is missing. It's really just practical, hands-on experience, and in particular, knowing how to not only sort of go through the mechanics of pipetting or sequencing or flow cytometry, but really training the mind to ask really important, great questions and to design experiments that actually will test a hypothesis and give you hopefully a quantifiable yes/no result at the end.
Spoken like a true scientist.
Oh, I agree. And one of my old mentors, Balfour Sarter, was an MD who then also got the science bug and never got the other degree, but he demonstrated, just like yourself, that you don't need it if you get it right, the training right in the end. So I know, Brenda, you are the resident cellular therapy person, so I assume you have about 1,000 questions here ready to go.
Oh boy, do I. Yes, I would like to talk, T cells. So adoptive cell therapy has been like on the making for decades now. In fact, Steve Rosenberg was one of the pioneers by using TIL therapy, so tumor infiltrating lymphocytes. But you're focusing on using peripheral blood derived cells mostly, and CAR T cells, also diving into receptor TCR transgenic T cells. So what does a cell have to do to become a therapeutic, and how do you choose the right cells? And how has your research focused on this question?
Yeah, so I think really great question. So first and foremost, it starts with having a T cell that is empowered and endowed to recognize a tumor cell. Ideally, recognizing a target that is highly enriched for or unique for cancer cells so that you can avoid on target but off tumor toxicities. So as I see it, it really just begins with finding a tumor reactive T cell, or if a tumor reactive T cell does not sufficiently exist within a patient, to engineer in that kind of reactivity using what we've done clinically most successfully to date has been, using integrating retroviruses. But there's some really exciting non-viral strategies that can also be used to not only introduce an antigen receptor, but really rewire a T cell to respond to its environment in completely new and novel ways. So step one is really either identifying or engineering T cells that have the right kind of reactivity. So I think there's no one solution to this problem, but in general, I think there are certain key requirements that you need to find in terms of the kinds of antigens. So first and foremost, especially if one is seeking to target solid malignancies, which is really the focus of my laboratory, the focus of my clinical work, you need to find targets that are almost exclusively expressed by tumor cells and not normal tissue. So this is a little bit of a different paradigm than the CD19 CARs, for example, which have entered the standard of care, not just in North America, but also in Europe. Moreover, I think it's quite clear, in really taking a page from our colleagues who use targeted therapies to drug constitutively active oncogenes, you need to find targets that are not just tumor specific, but also homogeneously expressed both within a tumor mass as well as throughout the body. And finally, you need to find targets that when you put them under immunologic selection pressure, it is very difficult for a cancer cell to lose expression of those targets and still be a lethal cancer cell to avoid potential immune evasion mechanisms.
On that line, you have now a publication by Archive in which you're looking into public neoantigens, which sounds a little bit like a paradox because it's really hard to find neoantigens that are shared among patients. And what's up with that? What is your view, your vision for the antigens that we're going to start targeting?
Yeah, so the focus of my group really has more recently coalesced around the idea that there may be a very interesting group of shared cancer-specific targets that are derived from recurrently mutated, in particular, driver oncogenes. And so I'm a bit of have some socialist tendencies, if you will. So I really like and embrace the title public. The goal of the public neoantigen concept is that there may, in fact, be a group of neoantigens that are not patient specific because they result from passenger mutations, but may be shared by groups of patients. And the reason they might be shared is because the epitope that is recognized is formed from recurrently mutated hotspot mutations in driver oncogenes. Now, the challenge, of course, is that these are intracellular proteins, and so these targets in general are not displayed directly on the surface of a cell. So you can't use antibody-based therapeutics or CAR therapies to target this class of antigens. You need to use an alternative antigen receptor, which is the T-cell receptor. And of course, the T-cell receptor, one of the, I tell my students, the TCR is, I'm very biased, of course, but is just one of the most magical entities, the most magical receptors in all of biology. And it's because it can do this incredible magic trick of looking inside with extraordinary efficiency to interrogate the intracellular proteome, but do so in a way that is displayed on the surface of the cell. And of course, the trick to this magic trick is HLA restriction. So in the case of the public neoantigens, the challenge is to not only identify such shared epitopes, but understand whether or not these are restricted by HLA alleles that have some common representation in the general population. Now, I think it's important to know a couple of important facts. First of all, this is a point I think we'll cycle back to. What is the definition of prevalent? And I think there are some enabling technologies that can really redefine how prevalent a particular HLA allele needs to be useful. Second of all, the notion or the premise that shared neoantigen reactivity is extraordinarily rare is really based on the observation that if you screen very disparate groups of patients who have heterogeneous diseases for reactivity that is contained in their tumor-infiltrating lymphocytes, or TIL, that if you cannot find reactivity against shared antigens, then it must not be common. But this presumes that there is a gold standard for reactivity. This presumes that TIL is the ultimate arbiter of what is reactive and not reactive. And there's some very, very elegant work, including work from a Dutch investigator, Ton Schumacher, who has shown that there are demonstrably immunogenic neoantigens, but that there is a failure for a myriad of reasons for a natural T-cell response to rise to the level of detection within TIL. But if that epitope is then, quote, outsourced to a broader repertoire from an unaffected individual, you can find reactive cells. So first and foremost, I think it's important for the field to recognize that the statement that there are not recurrently immunogenic neoantigens using TIL as the benchmark really suffers from a sensitivity problem, that TIL is really not necessarily the gold standard of what is and what is not immunogenic.
So to go along those lines and to kind of put on the practical hat with all the biotech I've been up to lately, if it's restricted to an HLA allele, for let's say you find the best public neoantigen you can for a cancer, or maybe a bunch of cancers all have the same one if you're really lucky, right? So this PI3K or PIK3CA, pick anything. What percent do you think, because these alleles are distributed across the population, how many percent of people with that cancer type or that scenario end up having the allele? Are these restricted to uncommon HLA types? Are they the most common ones that are going to pop up, and so you have a target? Do you imagine a repertoire where you're going to make a product that is for this public neoantigen, and then you make 8 of them for 8 different HLA types, or the ones that all work, and then that's how you FDA clear it, and so you get HLA typed before you get your therapy? Like I don't know if you've thought about much of these. I assume you have to an extent, but I'm thinking about that from kind of those perspectives.
Sure. So I think, again, immunologists and the cancer immunotherapy community should take a real page out of the experience of those who are involved in developing targeted therapies. So what the individuals involved in developing targeted therapies have come to realize is that the trick is really to screen patients in a very user-friendly way. So in the case of targeted therapies, it's really standard, not just at academic medical centers, but even in general practice, in the middle of America, in community-based practice, to the moment a patient is diagnosed with an advanced cancer, to send a piece of that tumor for next generation sequencing to identify potentially actionable genomic alterations that you could pair with a therapy. And in the case of certain recurrent fusions, such as a fusion called NTRK, these fusions are remarkably rare. They only occur perhaps in 0.5% or 1% of patients with cancer. But there has been the development of remarkably effective targeted therapies that target cancers with these recurrent alterations. And so if you can identify these patients and pair them with the right therapy, it has been utterly life-altering for these patients. Patients who had diseases that had expected life expectancy measured in months are now living many, many, many years just because the patient could be matched genomically with the right kind of effective therapy. So I think this really provides a very nice roadmap or template of what can be possible with, so-called targeted immunotherapy or public neoantigen targeted therapy. All of the seeds are there in our patients. We're already obtaining in the medical record the driver mutational landscape of our patients. All that is needed is understanding the patient's HLA haplotype. Well, for the vast majority of these next generation clinical sequencing approaches, a sample of normal unaffected tissue, most typically white blood cells just taken from the circulation, are taken and used as a reference genome. So that becomes the substrate already to elucidate a patient's HLA haplotype. And in point of fact, at Memorial Sloan Kettering, we have a clinical next-gen sequencing platform called IMPACT. This is FDA approved. This is CLIA certified. For research purposes, we are able to elucidate the HLA haplotype of our patients in real time. And there are other commercial entities. I'm not providing a commercial plug for any particular next-gen sequencing company, but a very large next-gen sequencing company in North America, Foundation One, also does targeted next-gen sequencing to look for actionable mutations, but also has the capability of elucidating a patient's HLA haplotype. So I think all of the seeds are there to develop these kinds of precision immunotherapies.
So in so much as your crystal ball can predict the FDA, do you think they're going to, I think they would buy, this only works for this HLA type. That seems reasonable. They'll do that now for anything. And they did approve the first immunotherapies for all microsatellite instability cancers. Do you think, and I think they will, but I was wondering your thoughts, do you think they'll go for like this is the neoantigen and these are the 3 different versions for the different HLA types that have it approach? Because I think that's where you're talking about going. You have the ability to measure it, but then tailoring that therapy, which also means the trials would be interesting too.
Yeah, throughout the world, there's already a regulatory template for so-called target-specific tissue-agnostic therapies. And in particular, these target-specific tissue-agnostic therapies take the case of, MSI-unstable cancer. Just by a single genomic alteration, you can give a patient an immunotherapy, and that will be reimbursed by insurance companies. So I think there's already the conceptual framework for that. I think what the challenge is envision a situation where you're reporting an HLA haplotype into the patient's medical record. If that information is used to generate and screen patients for a allogeneic stem cell transplant, perhaps the resolution of what's reported on these kind of tests are not the same sort of gold standard as might be done by the Red Cross or sort of dedicated high resolution sequencing. So I think there will be, maybe some disclaimers that are reported saying, you can't transplant someone based on this information, but you could use this information to determine if a patient might be eligible for a target specific but tissue agnostic TCR therapy.
Talking about tissue agnostic, what in your mind, what is the future of T cell therapies and adoptive cell therapies for the different types of cancers. I think a lot of the immunotherapy has benefited particular cancer types. Melanoma is probably the poster child of immunotherapy. But I know you're also interested in, for example, gynecological tumors. And what do you think is the potential of adoptive cell transfer to these other tumors that don't really respond that well to other types of immunotherapy? And I also want maybe to talk to our listeners about what are trucks.
So let's talk about the first question, which is what role will cell-based therapies, TCR-based therapies have for more common modestly mutated cancers? I actually think that the first TCR therapeutic that's going to receive regulatory approval is not a genetically engineered cell, but rather this really, really cool biotech construct, which is a recombinant soluble TCR that is a recombinant protein that is attached to an agonistic CD3 antibody, a so-called ImmTAC. And at one of the large North American cancer meetings, AACR, just a couple of months ago, it was reported a phase 3 clinical trial that used an ImmTAC, or a soluble bispecific TCR that recognizes a shared tissue differentiation antigen, GP100, in a group of patients who have a very, very modestly mutated cancer that has tremendous unmet medical need, which is uveal melanoma, or the rare subset of melanoma that's not highly mutated because it's associated with UV radiation, but rather starts in the back of the eye, in the pigmented cells of the eye, because of a recurrent, very potent set of driver alterations. So in this group of patients that really don't respond to PD-1, PD-L1, high-dose IL-2, it was shown that the company and the investigators were very, very brave. They designed a randomized trial so a physician could choose, anything in their armamentarium versus this experimental recombinant soluble TCR. And this trial showed an overall survival benefit, demonstrating that treatment with this TCR-based therapeutic could extend the life of patients with really treatment refractory, very otherwise poorly immunogenic subset of melanoma. So I think that just really portends that, number one, TCR-based therapies are imminently on the horizon. I don't know if this will be approved later this year or early next year, but I think on a timescale measured in months, we're going to live in a world where there are approved TCR therapeutics. And I think this provides a very important demonstration that TCR therapeutics are going to have a clear role in patients with very, very modestly mutated cancers that otherwise don't respond to easier forms of immunotherapy.
So talking timelines, you've talked about public neoantigens, which I'm getting back to. Where is that in the universe of clinical timeline? Is it, that nice pre-IND where they're starting to talk and things are moving? Is it still very academic? Yeah, I don't know if you can disclose that because of ongoing work, but I was kind of wondering where you kind of predict that is in the tube.
Yeah, so first and foremost, there have already been trials that have been initiated targeting groups of epitopes that meet the public neoantigen definition. In particular, a epitope derived from a recurrent mutation in KRAS G12V. For the PI3 kinase hotspot mutation that we describe in our manuscript, we have formed a partnership with a biotechnology company and are moving very, very aggressively. We're in the midst of IND enabling studies. And so hopefully by late 2022, we can begin enrolling a first-in-woman clinical trial that makes use of the TCR that we identified in our preprint.
Finding the right antigen to target is, of course, critical for these therapies. What I find really fascinating is also the effort that is put and the advances in the design of the receptors on the other side of the equation, and how very creative designs are coming into research, moving forward from the original CARs. And I think an example of this is what is known as truck T cells. Not to be confused, this is TRUC T cells, not to be confused with TRUCK T cells, which are T cells that produce specific cytokines upon CAR stimulation. But these other drugs have kind of harnessed the best of both worlds. They have the specificity of a CAR, but then the signaling of a TCR. Maybe you want to talk about a little bit about what it means to mix and match these different domains.
Thanks for the kind reminder. So these are being clinically developed by a biotechnology company called TCR Squared. So this can sometimes provide a little bit on first blush of confusion about, what is TCR Squared? What is a TRuC? Is it a TCR? Is it a not? So, as I was saying, TCRs is this incredible antigen receptor that has some seemingly paradoxical properties. One is that it is a relatively low affinity receptor compared with a monoclonal antibody. And yet, one of the amazing things about a TCR is that the entire complex can perceive as few as 1 to 3 molecules on the surface of a cell. And this contrasts with CAR, which appears, when this has been quantified, to require many thousands of molecules on the surface of a target cell in order to receive triggering. So what a TRuC receptor, it's a very clever design that actually attaches the antigen binding scFv to a component of the TCR signaling complex, one of the CD3 epsilon molecules. And this interdigitates into the full TCR complex. And the upshot, the functional upshot, is that a TRuC modified cell still makes use of a monoclonal antibody derived binding domain to recognize its target. So it's HLA independent, but recruits all of the TCR signaling complex. And so it can perceive antigen density that is several orders of magnitude smaller than what is conventionally required for a CAR. And I think this is so cool and so important because of a common mechanism of acquired resistance among CAR therapies is that the tumor cell does not have to lose total expression of the target antigen. It can just have a relatively modest decrease in expression of the target antigen below the threshold of a CAR, which again is several thousand molecules at least. And that becomes basically invisible to a CAR modified cell. Theoretically, a TRuC might be able to see, many, many fewer molecules, and therefore that pathway of resistance would be reduced. But these trials are ongoing right now.
It's very interesting also in the field of synthetic immunology, what kind of signaling molecules you can combine to modify the function of these cells, because starting from the first generation of CARs, which were fairly simple, there's a lot of combinatory combinations of receptors with different types of signaling. So it's fascinating. It's just a matter of having creative and looking at the right components.
Yeah, and I think that really it's an incredible opportunity, but the combinatorial diversity that can be generated, especially because you can kind of mix and match, what's on the extracellular domain, what kind of signal is transmitted to the inside of a cell or blocked on the inside of the cell. This just multiplies in a hugely, geometric way. There are so many permutations. It gets really challenging trying to empirically pick the best one. This is just a shout out. I am such a fan of my friend and colleague, Alex Marson at UCSF. Alex has been a champion for several years now of, we were talking earlier, the standard way of integrating, new payloads, including antigen receptors into T cells is integrating viruses. Alex has really championed and innovative non-viral strategies to put antigen receptors in. And more recently, what Alex and his team have been able to do is to integrate these synthetic receptors or switch receptors that can sort of mix and match extracellular and intracellular domains and put these into pooled biologic screens and really let the best sort of T cell modification win by different parameters. So I think these sort of large combinatorial screens where different combinations are in effect raced against one another is really charting the path to the future.
So do you envision a almost like a hot swap platform that may exist some point where you can, swap, you can almost get like a platform approved and then swap out either antigen, part of the receptor on one end or the other to like, this is the therapy that's validated and these are the targets, and you swap one another. We're seeing this now, like with the idea of the Moderna vaccines and these mRNA vaccines that, theoretically the next antigen put in them is easier to prove. But I was wondering if you can see this as well, maybe even for CAR Ts or other types of, synthetic biology receptors.
So let me sort of divide your question into two different areas. So first, let's go back to sort of the notion of pooled screens, the notion that there's huge combinatorial diversity. There's a lot of sort of tweaks and switches we can apply to T cells. Establishing clinically which is the best one is just untenable, right? We never, these are such bespoke, challenging therapies to manufacture, to deliver. The idea that we would do an endless series of, what currently is the gold standard of randomized trials, half the patients get modification X, half the patients get modification Y, and you compare those two, is simply untenable. So one idea, acknowledging that limitation and challenge, that I've become really enamored with, and this really extends from, my background and love for syngeneic mouse modeling, is the idea that in mouse models, using conjugate markers, right, mouse cells that have allelically identifiable cell surface markers, that when you co-infuse those into a mouse, you can really do competitive repopulation experiments and ask, which cell population with particular genetic alteration leads to a phenotype in vivo that might be desirable. And I think that has been such a powerful tool among, syngeneic mouse modeling. Why can't we apply that same concept to human beings? Why can't we, by design, infuse patients with genomically distinct, well-characterized but mixed populations, and effectively race these different populations in a competitive repopulation experiment inside of the patient, and ask the question, if the question is, for example, which genomic perturbation leads to enhanced T cell trafficking to tumors or enhanced T cell persistence, these kind of unique clinical trial designs could very, very efficiently, in just a few number of patients, really quantitatively test these hypotheses, address that question, and then we can move on and say, okay, this intervention did not lead to enhanced T cell trafficking, but this did. We can incorporate that into subsequent therapies. So I think that's sort of one innovative trial design that I think is underutilized and I'd like to see a lot more of. Jason, to more directly address your question, I think there's a precedence, especially with these non-viral integration strategies, because these in general make use of very targeted insertions, using homology-directed repair. I think there's a precedence already where what the regulators really want to demonstrate as being safe is the genomic integration, the homology arms, the locus that is being integrated into. But if, for example, you're taking the unmanipulated variable sequences of a T cell receptor that has already been inside of a patient that did not have toxicity, to basically use those inner bits, those unique variable bits, in a modular way so that you could mix and match. You could, for example, take TCR clonotypes from a patient that had patient-specific or private neoantigen responses, but is not having a response to just that alone. Or you could use that as a way of mixing and matching these public neoantigen TCRs into patients in a very defined way.
It's exciting to think about this, and we think about the technology, and I think it's important to realize like how much of this is predicated on things like CRISPR technology or even PCR that like, I remember when I started doing PCR, doing real time was like extra super fancy. And like you still did it on a gel. And I know even before my time, it was in a water bath, and it wasn't much before that, before they didn't even have it. And so just thinking about like, that wasn't that long ago, really. Like maybe not even 30 years really in a lot of ways. And now because you have that, you can do, T cell directed therapies and, targeted therapies. It's kind of humbling.
You could do pooled genomic screens inside a human patient. That's wild, right? And I think we're really, technologically, we're empowered to do that.
There's so many advances, including, for example, CRISPR, right? What you mentioned, the lab of Alex Marson, they're doing amazing directed targeting of particular TCRs. It can introduce anything on a T cell. It's fascinating to see how far and how much the sky is the limit, or the efficiencies of integration are the limits, but they're working on it very, very efficiently also. It's been such a great conversation, and I think that our listeners have learned a lot about the promise, the story, and what T cells can do for therapy of cancer and also other diseases as well. But in a more kind of personal note, we like to ask you, we always ask a question at the end of the podcast to our scientist guests. But if you were not a scientist, what would you be?
That's such a great question. I think I'm suffering here a little bit from recency bias. I really enjoyed watching the Tokyo Olympics. I think this was a fantastic form of escapism from COVID-19. And it turns out this was the first year that skateboarding was a medal winning event in the Olympics. And Japan, incidentally, just destroyed both men's and women's skateboarding. So when I was younger, I just took so much pleasure in riding my skateboard. And so I think there's a piece of me that never really lost the skater mentality. And so I think if I could do anything, at least the idea of being a pro skater sounds pretty cool right now.
I'm afraid the sidewalks in New York are not good enough for skating around, are they?
Actually, at Memorial Sloan Kettering, there is a ledge which some local skateboarders love to use. They can do grinds, they can do slides on it. And so I'm in my office and I hear them skateboarding, and have befriended them. So even in New York City, there's some great skate spots when the weather permits.
So TikTok video with you doing a kickflip, right? 6 months.
On that note, I think that the youngest ever gold medalist was it a British girl of 13 years old on a skateboard?
Yeah,
I think I heard that news.
Sky was her first name. She had some great runs.
It's been a pleasure. Thank you so much for joining us.
Thank you, Brenda and Jason. My pleasure also.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup papers. You can also reach out to us on Twitter at Immunopodcast or via email at info@immunologypodcast.com with feedback or to suggest guests. See you next time.